welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Biomolecules, and we are in part 3 of the series. So if you haven't checked out part 1 and 2, please do so. The links will be there in the description box below. Alright, so let's begin. Okay, so question number 1. Adenine is A. Purine, B. Pyrimidine, C. Nucleoside, or D. Nucleotide. Okay, and the question appeared in the year 1992. So adenine is a base and it's a purine base. Okay, so the answer is A. Let's go to the discussion. So adenine is a purine base and it is present both in RNA and DNA. Alright, so and this is the structure of adenine. Okay, so adenine is one type of purine base. There's guanine, which is also another purine base. Alright, it's a type of purine base. Alright, so below you see the structure of adenine. Okay, this was a pretty direct, simple. Alright, so let's move on then. Question number 2. A nucleotide is formed of, and this appeared in the year 1991, A. Purine, pyrimidine and phosphate, B. Purine, sugar and phosphate, C. Nitrogen base, sugar and phosphate, D. Pyrimidine, sugar and phosphate. So nucleotide is formed of, it can be purine or pyrimidine, so it's a nitrogenous base, right? So it has to be C, nitrogen base, sugar and phosphate. Okay, so the combination of all three forms a nucleotide. So the answer is C. Let's go to the discussion. So nucleotide is formed of purine or pyrimidine nitrogenous base, ribose or deoxyribose sugar, and phosphate. Okay, phosphoric acid, which is attached to it. Okay, so purine or pyrimidine base, you know, it could be adenine, guanine, thymine, cytosine, or uracil if you are talking about RNA. Okay, this is for DNA, this is for RNA. Okay, along with that, it should be attached to either a ribose sugar or deoxyribose sugar, again based on what we are talking about, whether it is RNA or DNA. So ribose sugar appears in RNA, deoxyribose sugar is in DNA. All right, and of course the common factor in both is the phosphate molecule. Okay, that's the common factor in DNA and RNA nucleotide. All right, so and this is very important to remember. What if it's a nucleoside? Then what do you say? Okay, if it's a nucleoside, then it will only contain first two. Okay, either it will be ribose or deoxyribose sugar plus the nitrogenous base. Okay, this combination forms nucleoside. These three together form a nucleotide. Okay, the difference between nucleoside and nucleotide. Remember, nucleoside comes first, then followed by nucleotide, where the phosphate is attached. Okay, clear? I guess we've answered the question. Yeah, this is also pretty simple, pretty direct. Let's move on. Question number 3. DNA is composed of repeating units of, and this appeared again in 91, A. Ribonucleosides, B. Deoxyribonucleosides, C. Ribonucleotides, and D. Deoxyribonucleotides. Again, we just discussed the difference between nucleotide and nucleoside. Now, when we're talking about DNA, okay, DNA is composed of repeating. Now, it's a deoxynucleic acid, right? It consists of repeating units of what? It will be D. Deoxyribonucleotides. Okay, it will be nucleotides. All right, so the answer here is D. Okay, yeah. So it's composed of repeating units of deoxyribonucleotides. This structure is taken from NCERT, and clearly you can see just one part of the DNA. They're just showing you the different bases, thymine and adenine and guanine, cytosine, and how they are attached to each other, right? They form here, adenine and thymine form double bond, over here guanine and cytosine form triple hydrogen bond. All right, you can see the phosphodiester bond over here. Okay, now remember, you need to remember the types of bonds that are there, okay, in a DNA. But what I want to convey here is uh, how the DNA consists of deoxyribonucleotides, okay. So here you see a segment of the DNA, okay. So here you have the deoxyribo sugar, all right. So you have the phosphate molecule here, and of course you have the nitrogenous base over here, okay. So you have all three present there. So yes, of course they are nucleotides and they are repeating units of nucleotides, all right, in a DNA. Okay, all right. So again, this was a pretty direct, simple. Let's move on then. Okay, so question number 4. The basic unit of nucleic acid is, and this again appeared in 91, basic unit of nucleic acid. Okay, so A pentose sugar, B nucleoid, C nucleoside, and D nucleotide. Of course, it's D nucleotide, the repeating units of nucleotide, okay, whether it's DNA or RNA. Okay, and this is like another form of the previous question. So the answer here is D. So, one second. Nucleic acids are repeating units of nucleotides, also known as polynucleotides. Of course, poly means there are more than 2 or 3 nucleotides that are continuously being repeated. Sorry, comes to the next question. So yeah, that pretty much answers the question here. The basic unit of nucleic acid is nucleotide, right? Let's move on then. Question number 5. RNA does not possess, this appeared in 1988, pretty old question. RNA does not possess A. uracil, B. thymine, C. adenine, or D. cytosine. So it does not possess. Please read the question properly. It does not possess B. thymine. Don't just like read the question quickly and then go to A and say uracil. Yeah, RNA does possess uracil. We know that, but it does not possess thymine. All right. So the answer is B. So the bases in RNA are adenine, guanine, uracil, and cytosine. Thymine is absent in RNA. Very, very important information. You should remember this. Uracil is present in RNA and thymine is absent in RNA. All right. So let's move on. Question number 6. A Ramachandran plot is used to confirm the structure of A. RNA, B. proteins, C. triacylglycerides, and D. DNA. It's appeared in 2019. So Ramachandran plot, if you are not aware, and it's something that you will probably study later on in college, is used to confirm the structure of proteins. All right. Answer is B. So remember this as a fact because this is something that you study in college. Maybe not too much, not really much mentioned here in school level. Ramachandran plot is used to determine the spatial arrangement of polypeptide chains in proteins. Okay, so now polypeptide chains in proteins are arranged as primary structure, secondary structure, tertiary structure, quaternary structure. Especially when you look at the secondary structure or the tertiary structure, you'll find many hidden bonds in the protein. Okay, which attach the polypeptide chains together. Okay, and they are arranged in a certain way. So Ramachandran plot is a software where you can actually see or assess the structure of a particular protein, how they are arranged, what are the different bonds that are there inside the protein, how are they all arranged inside. So yeah, Ramachandran plot helps you to understand that. Okay, like I said, remember this as a fact. Yeah, for exam. I don't know if they'll ever ask you, but yeah, if they do, you know the answer, right? It's used for understanding how proteins are structured. Okay, let's move on then. Question number 7. Which of the following is the least likely to be involved in stabilizing the three-dimensional folding of most proteins? This appeared in NEET 2016. A. Hydrogen bonds, B. Electrostatic interaction, C. Hydrophobic interaction, D. Ester bonds. Like I previously mentioned, when you look at the tertiary structure, you'll find many bonds. The hydrophobic bonds, the hydrogen bonds, electrostatic interactions are also there. There are many bonds that are there that are holding the protein together. and of course stabilizing them, right? So they don't break off or they get denatured. So you need the protein structure to be stable. So of course there are many bonds that are present inside that protein. Okay, so now which is the least likely to be involved? Okay, the least likely to be of course we know there are hydrogen bonds present in proteins that help them stabilize the structure. There are electrostatic interactions, yes. There are hydrophobic interactions, yes. But ester bonds is, least likely to be present in a protein. Okay, that helps in stabilizing its structure. The ABC, yes, they are present there, but ester bonds, no, we don't see that in proteins. All right, so the answer is B. So the three-dimensional or 3D arrangement or tertiary arrangement structure of proteins contains hydrogen bonds, they contain disulfide bonds, they contain ionic bonds, electrostatic bonds, and hydrophobic bonds. But there are no ester bonds to stabilize the tertiary arrangement of proteins. Okay, so yeah, like the question said, which is least likely to be present? Of course, it's ester bonds. Okay, and yeah, where else do you see ester bonds? You see ester phosphodiester bond in the DNA, RNA. Yeah, so that's one place where you see that. So there, ester bonds are helping in stabilizing the structure, but not in proteins. So the question, of course, related to proteins, and of course, there are no ester bonds there. Okay, please remember this also. This is a very good question. Okay, it's a very good question. It wasn't just direct. You had to think. All right, so let's move on then. Question number 8. The figure shows a hypothetical tetrapeptide portion of a protein with parts labeled A to D. Which one of the following options is correct? This appeared in NEET 2013. So you have A, D is the acidic amino acid glutamic acid. So you have D here. But the structure here is not that of a glutamic acid. Okay, the structure here is not that. B, C is an aromatic amino acid tryptophan. No, that is also incorrect. A is the C terminal amino acid. No. And D is the N terminal amino acid. No, again that's incorrect. A is a sulfur containing amino acid methionine. A, no, that's also incorrect. Actually, there's a problem in this answer because there are no correct answers in this. So even if you attempt it, I guess you'll get some bonus marks for it. But yeah, I guess they made a mistake in the question itself, right? So, but we'll go to the discussion and see what could be the possible answers there. So C is aromatic amino acid tyrosine. A is N terminal amino acid and D is C terminal amino acid. It was the other way around. And B is sulfur containing amino acid. It is cysteine. Okay. If you go back to the question, yes, there's sulfur here. It says A is a sulfur containing, no, because there's no sulfur in A, right? So clearly it's not A. So it's B is a sulfur containing amino acid. It's not methionine, it's cysteine. And here it's likely not, it's not glutamic acid. I don't know, the structure of this amino acid is actually incorrect. I think they wanted to print it as glutamic acid, and probably option A could have been the right answer, but they have made a mistake in the structure. It's not glutamic acid. So there's a problem here. But the others are pretty explainable. Uh, yeah, like I said, A is a sulfur containing. No, that's definitely not. A is not a sulfur containing amino acid. It's B and it's cysteine. It's not methionine. And A is a C terminal amino acid. No, it's not. It's N terminal amino acid, and this is C terminal amino acid. So this should have been interchanged. And C is aromatic amino acid tryptophan. No, it's tyrosine actually. Okay. Oops, sorry. Let's go back. Yeah. So here you see the sulfur containing amino acid, which is cysteine. Okay. All right. Let's move on then. Question number 9. Identify the substances having glycosidic bond and peptide bond respectively in their structure. This appeared in NEET 2020, pretty recent. Okay, so which one of them has a glycosidic bond and a peptide bond? So A, chitin, B, this, I mean, cholesterol is the other option there, but no, cholesterol does not have a peptide bond. B, glycerol, trypsin, no, that's also incorrect. C. Cellulose, lecithin, no. And D. Inulin, insulin. It's actually D. Inulin is saccharide, carbohydrate, and insulin is a protein. Okay, and both have peptide bond. Sorry, insulin has a peptide bond and inulin has a glycosidic bond. Right? So the answer is D. Let's go to the discussion. So inulin is a polymer. It's a polysaccharide of fructose units, that's B2 and 1 linkage, with a glucose molecule at the end of each fructose chain. The glucose molecule is attached to the fructose chain by alpha 1,2 linkage. Alright. Remember this structure of inulin pretty much comprises of polymers of fructose, but at the end of the chain they have glucose. Okay, that's how inulin is structured. It has low molecular weight, so it's easily digestible, it's easily soluble in water also, and these linkages are formed by glycosidic bond. Okay, in insulin, now please don't confuse inulin with insulin. Okay, what if the options were the other way around? Okay, what if glycosidic bond, it said insulin, and peptide one said inulin? Okay, that's also incorrect, right? So many people get confused between inulin and insulin. Inulin is a polysaccharide and insulin is a protein, it's a hormone. Okay, secreted by the pancreas. Insulin is a very important hormone in our body. I've already discussed this in much detail, the functions of insulin and everything in endocrine system, so please check that out. So insulin is a hormone secreted by the pancreas and is protein in nature. Okay, it is made up of 2 polypeptide chains held together by disulfide bridges. Okay, so this is basically the structure of insulin. So there are various steps by which the final structure of insulin is formed. You have pre-insulin, which is then converted to the final structure of insulin. Okay, it has 3 chains, and then finally it comes down to 1 chain. Sorry, 2 chains. I'm sorry, 2 polypeptide chains. Okay. So yeah, the amino acids are held together by peptide bonds. Of course, it's a protein, so there are amino acids, and these amino acids are held together by peptide bonds. Okay, so here I've put the structure of inulin. Okay, you see inulin here. You see this fructose. I'm sorry. Let me get rid of this. I think my computer is going crazy. Yeah. See the structure here. This is fructose chain. This is the glucose that's attached to this fructose chain. Okay, this is pretty much the structure of inulin. Now what you see here is insulin. Okay, what you see here is insulin. Okay, you have A chain, B chain, there are disulfide bridges. Of course, these are all the amino acids that are attached to each other, but they're just represented as a line, but they're actually amino acids that are attached to each other. And of course, these amino acids are attached via peptide bonds. So here you see a diagram of peptide bond. Okay, an example. This is just an example of how a peptide bond is formed. Okay, between the carboxyl end and the amino end, and then water is given out, and yes, you find this peptide bond here. Okay. So yes, this is what inulin is and what insulin is. Okay, so if we go back to the question, clearly the other options did not make sense. Yeah, cellulose, no. Lecithin, no, it can't be that. Glycerol, no, there's no glycosidic bond. Trypsin, no. Trypsin, yeah, the peptide bond is there, but yeah, glycosidic bond is not there in glycerol. So trypsin is an enzyme, right? So yeah, glycerin is phosphoglyceride, no. Cellulose, yes, there's glycosidic bond here, but it doesn't match the option. Chitin, cholesterol, no, again doesn't match the options there. Although chitin has glycosidic bond, cholesterol is a lipid, so there's no peptide bond there. So yeah, obviously if you go through A, B and C, obviously option D is the appropriate answer. It's inulin and insulin. Okay, let's move on. This was again a good question. deserves a smiley face. Okay, let's move on. Yeah. Question 10. Which of the following biomolecules does have a phosphodiester bond? Okay, this appeared in 2015. We just discussed phosphodiester bond. A. Amino acids in a polypeptide. No. B. Nucleic acids in a nucleotide. C. Fatty acids in a diglyceride. B. Monosaccharides in a polysaccharide. So phosphodiester bond actually is nucleotide in a nucleic acid. I think they've framed it incorrectly. It should have been the other way around. Yeah. So nucleotide and nucleic acids have phosphodiester bond. How they are connected with each other. They're repeating units of nucleotides in a nucleic acid, right? The others A, C, and D are incorrect. So the answer is B. Let's go to the discussion. So nucleotides and nucleic acids have phosphodiester bond. That's how they are connected with each other to eventually form nucleic acids. That's RNA or DNA. All right. Okay. This was also pretty simple. This was direct. Yep. Let's move on then. Okay. So question number 11. Which is wrong about nucleic acids? This appeared in 1993. A. DNA is single-stranded in some viruses, which is correct. B. RNA is double-stranded occasionally. Yes. Length of one helix is 45 Armstrong in bDNA. No, this is wrong. So actually C should be the right answer. D. One turn of zDNA has 12 bases. Yes, that's correct. So the answer is C. So in bDNA, now there are there's zDNA, there's bDNA, which is present in most living systems. So what we study is structure of bDNA. The Watson-Crick model that we study, which you will probably study in genetics, right, is actually bDNA. Okay, and the length of one helix, that means one curve. Okay, one curve, like if you have a double-stranded DNA, so one curve, okay, or one helix, like they say. So this is a helix or a turn. They call it turn. Now this angle here, the length is 34 Armstrong. Okay, the length of this helix is 34 Armstrong in bDNA. Okay, remember it's bDNA, and this one helix has 10 base pairs. All right. This one helix has 10 base pairs, and the length of that helix is 34 Armstrong. Basically, that's what it is. Okay, the other option that was D, one turn of zDNA. Change this. zDNA has 12 bases. Yes, that's correct. Okay, one turn of zDNA has 12 bases. But yeah, what is wrong about nucleic acid? Length of one helix is 45 Armstrong in bDNA. Okay. Alright, I guess we can move on. Question 12. A segment of DNA has 120 adenine and 120 cytosine bases. Alright. The total number of nucleotides present in the segment is, this appeared in 91. So you have a segment of DNA, it's double-stranded. So you have 120 adenine and 120 cytosine. Okay, now we know for sure that adenine bonds with thymine and guanine bonds with cytosine. So you have 120 adenine, you have 120 cytosine, right? Now the total number of nucleotides present in the segment is, of course, DNA has nucleotides, they have a deoxyribose sugar, they have a phosphate molecule, and of course they have a base where these connections happen, right, between the two strands. So you have 120 adenine, so clearly there'll be 120 thymine. You have 120 cytosine, so clearly there'll be 120 guanine, and they will form nucleotides. So the total number of nucleotides present in the segment is the total of all 4, okay, which will be 480. So you add all of them up, you will get the answer D, which is 480 nucleotides present in the segment. So adenine bonds with thymine, cytosine bonds with guanine. Just explain that, and your final answer should be 480. Okay, number of nucleotides in the segment would be 480. Okay, again, this was direct. Basically, they were just trying to test you on what the structure of DNA looks like and what is the difference between bases and nucleotides, how are they formed, and which bonds with what. So, this was basically a test on that. Okay, let's move on then. Question number 13. Which is not consistent with double helical structure of DNA? It's been 1990, pretty old question. So, you have to find what is not consistent. A bonds with T, C bonds with G. Yes, that's consistent. Density of DNA decreases on heating. Yes, that's actually correct. So the answer B is incorrect as for this question. C, A plus T over C plus G is not constant. Actually, it is constant. So this is not consistent. Both A and B, no. So the answer is C. Now what they're really talking about is Chargaff's rule. Okay, which we will come to in the discussion in some time. I was going to say description for some reason. So Chargaff's rule proposed that the number of adenine was equal to the number of thymine. Right? We know that. Yeah, in a DNA structure, you have two strands. So adenine with thymine and cytosine with guanine. Right? Hence, in DNA, now DNA varies as in number of adenine or purines will be always equal to the number of pyrimidines that it associates with. But obviously, the number of adenine can be different from the number of cytosine, right? So, if you have, let's say, you have a DNA segment or whatever, and you have 120 adenine, but you have 50 cytosine, yes, that's possible for a particular species. But the ratio of that species will remain constant. Okay, it can vary from species to species, but it will remain constant for one particular species, this ratio. Okay, now this is what Chargaff stated. Okay, yes, the number of pyrimidines will be equal to the number of pyrimidines, right? But it's not necessary that A and purines and as in adenine and guanine, these two purines or these two pyrimidines will be equal in number. Of course, this can be 120, this can be 300. Yes, so but you'll have 300 guanine and you'll have 120 thymine. The ratio will always be 1, right? But when you are comparing A plus T over G plus C, this ratio, this will always be constant. Okay, this will always be constant for a particular species, right? This will always be constant for a particular species. Pyrimidines over pyrimidines, if you take the ratio, it will always be 1. Right, now A over T, if you take the ratio 120 over 120, will always give you 1. Okay, if you take a ratio of G over C 300 by 300 will always give you 1 or whatever it is, but that can vary from species to species. Okay, but if you take one particular species, you take humans or anything like that, but that will always be constant, A plus T over G plus C ratio. Okay, this will always be constant. If you add 120 plus 120, that will be what, 240, and if you say 300, 600, this particular ratio will always be constant for a particular species, but it will vary from species to species. If you just take one species into account, it should be constant over there. Okay, okay, clear? So number of pyrimidines will always be equal to number of pyrimidines, but when you take this ratio A plus T over G plus C, that will more or less be constant, but can vary from species to species. Yeah, so which is not consistent, of course, A, B and D were incorrect. C is the right answer. Okay, let's move on. Question number 14. In double helix of DNA, the two DNA strands are, and this appeared in 1988, A coiled around a common axis, B coiled around each other, C coiled differently, D coiled over protein sheath. Okay, so the two DNA strands are coiled around a common axis. Okay, so the answer here is A. So when you look at the double helical structure, so this is a very good diagram of the double helical structure. You can see a sugar here, you can see the base here, you can see the phosphate here, okay, forming bonds, okay, forming nucleotide bonds. And you have this double helical structure. Okay, this is a 5 end, this is a 3 end, this is 3 end, this is 5 end. So they are anti-parallel to each other. They are not parallel. So the 5 and 3 end will not always be like this. No, that's incorrect. Okay, so when you look at a double helical structure, this is 5 and this will be 3 end. They are anti-parallel to each other. Okay, but when they are forming this helix, okay, when they are connected with each other, as you can see this red line and this black line, there is a common axis in between, okay, around which they form a double helical structure. Okay, that's how they are attached to each other. Okay, so the two DNA strands in a double helical structure are coiled around a common axis. Okay, clear? So it's pretty simple. But you don't understand the structure of DNA first. Okay, let's move on. Okay, so question number 15. ATP is, this appeared in 2000, A nucleotide, B nucleoside, C nucleic acid, or D vitamin. Okay, so ATP actually is a nucleotide. So the answer is A. Let's go to the discussion. So ATP or adenosine triphosphate, that's the full form of ATP, have three phosphate molecules attached to ribose sugar. So you have three phosphate molecules that are attached to the ribose sugar, and I forgot to add there is an adenine. Nitrogenous base. Okay, there's adenine, nitrogenous base. Okay, that you can see here. So you have adenine attached to the ribose sugar, and you have the ribose sugar attached to three phosphate molecules. Okay, now the second and the third phosphate of ATP are attached by high energy bonds. So ATP is also known as like the energy molecule of our body, which gives us energy to perform functions, right? It also performs various activities, cellular activities also in our body. So when we look at the structure of ATP, of course we see that it is a complete nucleotide structure. Okay, it's a complete nucleotide structure. You have the ester bond that is formed here, but now these two phosphate molecules that are there, these are high energy bonds. Okay, these are what give off the energy to us to perform our functions. Okay, so the second and third phosphates of ATP are attached by high energy bonds. But ATP, if you look at the ATP structure as such, it will be considered as a nucleotide. Okay, it's not a nucleic acid or nucleoside or anything. It's a proper nucleotide. That's how it's classified. It's not nucleoside, it's not a vitamin, it's not a nucleic acid, it's a nucleotide. All right, let's move on. Question number 16. Match the following. This appeared in NEET 2020, pretty recent. A inhibitor of catalytic activity is malonate, possesses peptide bonds, collagen, cell wall material in fungi is chitin, secondary metabolite is ricin. Okay, so inhibitor, that's A2, okay, B4, C3, yep, and D1. So the answer is A. Okay, so malonate or malonic acid, now pretty much we've matched the following, the column A and column B, but I thought I'll discuss inhibition of enzymes. All right, so a malonate or malonic acid is a type of inhibitor. It's an enzyme inhibitor. Okay, and it's a competitive inhibitor. Okay, it's a competitive type of inhibitor, and it competes with substrate succinic acid to occupy the active site in the enzyme succinate dehydrogenase. Okay, so now if you look at this diagram here, in case you are not aware of what inhibition of enzymes is, let's say you have an enzyme, and enzymes have active sites, right, to which the substrate attaches to. Okay, so you have a substrate, you have the enzyme, it forms an enzyme substrate complex which eventually forms product. It can be a reversible reaction also, yeah. So substrate plus enzyme forms a transient state called enzyme substrate molecule, and then which then gets converted to a product. Okay, the final product that we see. That's how a reaction happens between substrate and between enzyme and substrate, which finally forms a product. Now there are some inhibitors. Okay, there are inhibitors that can stop the enzyme from performing its function. Okay, to help convert the substrate to a product. Okay, now these can be non-competitive inhibitors, they can be competitive inhibitors. We are particularly talking about malonate. Now malonate competes with the substrate. You have an enzyme, you have an active site. Okay, you have an enzyme, you have an active site over here to which the substrate will attach to form an enzyme substrate complex. Okay, and then there is making and breaking of bonds happening inside that, and eventually forms the final product. But now malonate is a type of inhibitor. That means it is stopping the substrate from attaching to the active site of the enzyme. That means it does not want the substrate to convert to the product. Okay, that's what basically uh, the function is. Okay, that's what malonate has to do. Okay, it's like an inhibitor which will stop the production of conversion of that substrate to a product. Okay, so now what it does is, now malonate is a structure that is similar to the substrate. Now, for example, a very good example is of the enzyme succinate dehydrogenase. Okay, succinate dehydrogenase we know is an enzyme, right? Its substrate is succinic acid. Okay, now if you look at the structure of malonic acid and succinic acid, the structures are almost similar. Okay, the structures are almost similar. It's a very interesting thing, study in nature, how you can create, or how nature is able to stop these reactions by creating something that is almost similar in structure and can fool the enzyme to thinking that I can convert it to a product, but I can't do it now, so I'm stuck. Okay, so it will attach into that pocket, into the active site pocket. Okay, substrate, I'm sorry, the competitive inhibitor can attach itself into that pocket. Okay, the enzyme thinking that it's a substrate, but it is not a substrate, it's an inhibitor. Okay, so now once the inhibitor has attached itself to the active site, it cannot get converted to the product. Okay, it is a inhibitor, it's an enzyme inhibitor complex which is formed. Okay, so it is competing with the substrate for that active site. Okay, there are active sites that are present on the enzyme. The inhibitor is actually competing with the substrate for that position, for the active site position. Okay, so basically this is what a competitive inhibitor does. Okay, it competes with the substrate, all right, for that same site. Okay, so malonate, like I said, succinate dehydrogenase is a very good example, where malonate or malonic acid is a type of competitive inhibitor for the substrate succinic acid. There are other versions of these inhibitors that can attach to succinate dehydrogenase. Other than malonic acid, there's oxalic acid, there's glutaric acid. They're also similar in structure with succinic acid. So they are very good inhibitors. Okay, they're very good inhibitors that can attach to the active site of the enzyme. Okay, and can stop the reaction from happening. All right, so I thought I'll discuss what competitive inhibition is, since we are talking about malonate or malonic acid in this, and that you can understand what competitive inhibition is. If we go back to the question, yes, one more thing, cell wall material in fungi, please remember, yes, chitin is one of the products that are present in the cell wall of fungi. Okay, secondary metabolite, yes, all these names are present in NCERT. I already discussed this in the previous video, so please check that out. Okay, and collagen we've already discussed multiple times, shouldn't be new to you. Let's move on. Question number 17. Consider the following statements. This appeared in NEET 2019. A coenzyme or metal ion that is tightly bound to enzyme protein is called prosthetic group. No, that's incorrect. A complete catalytic active enzyme with its bound prosthetic group is called apoenzyme. That is also false. So if you have to select the correct option, both are false. Both A and B are false. Answer is D. Let's go to the discussion. I think I already discussed cofactor, coenzyme, prosthetic group, all that previously, right? So coenzymes are non-protein organic factors that are loosely bound to the enzyme. Okay, they're non-protein. Now, cofactor is the main guy. Okay, you have cofactors. There are coenzymes and cofactors. There are prosthetic groups in cofactors. I remember saying this in the previous lecture. These definitions are very, very, very important. Like they could ask you something like this, and you are expected to answer. So if you don't know the definition, if you knew the definitions, it's very easy. Okay, so coenzymes are non-protein organic factors that are loosely bound to the enzyme. Prosthetic group is non-protein organic cofactor that is tightly bound to the enzyme. So enzyme has a protein part, has a non-protein part, right? And there are cofactors. It can be organic cofactor, there can be an inorganic factor which are called activators, the metal ions, okay, also play an important role. Okay, and coenzymes are loosely bound, prosthetic groups are tightly bound. Okay, they're tightly bound there. Remember this. Metal ions are inorganic, also known as activators, which attach to the active site of enzymes and enhance enzyme activity. All right. So these three definitions are very, very, very important. Also, holoenzyme is the complete enzyme containing an apoenzyme and coenzyme. Okay. So, yep, you have, let's say, holoenzyme is like just think of apoenzyme is the protein part of the enzyme. Holo is like the complete structure. It has the non-protein part as well as the protein part. All right. So think of holoenzyme as like the complete structure of the enzyme. Okay, that's holoenzyme. I guess I've answered the question. Let's go back to the question. A complete catalytic active enzyme with its brown prosthetic group is called apoenzyme. Yes, it can be prosthetic group, it can be coenzyme, it could be anything else, the non-protein part basically. Coenzyme or metal ion that is tightly bound to the enzyme protein is called prosthetic. No, that's also incorrect. That's an incorrect statement. Yeah, so this is what it is pretty much. Let's move on. Question number 18. Prosthetic groups differ from coenzymes in that this appeared in NEET 2019. Again, a similar question. Like I said, this topic is very, very important. You need to understand the difference between prosthetic group and coenzymes. A, they require metal ions for their activity. A, prosthetic groups are tightly bound to apoenzymes. C, their association with apoenzymes is transient. D, they can serve as cofactors in a number of enzyme catalyzed reactions. Prosthetic groups differ from coenzymes. It's clearly B, they, that means the prosthetic groups are tightly bound to apoenzyme, that means the protein part. So the answer is B. We just discussed this in question 17. So it's, So it's pretty much the same thing. Prosthetic group is non-protein organic factor that is tightly bound. Remember, prosthetic group is tightly bound to the enzyme, to the protein part of the enzyme. That's the apoenzyme part. Okay, let's move on. Question 19. Which of the following statements is correct with reference to enzymes? And this appeared in NEET 2017. A, holoenzyme is equal to apoenzyme plus coenzyme. B, coenzyme is equal to apoenzyme plus holoenzyme. No. C, holoenzyme is equal to coenzyme plus cofactor. No. D, apoenzyme is equal to holoenzyme plus coenzyme. No. It's clearly A. We just discussed this. Holoenzyme is equal to apoenzyme plus coenzyme. The answer is A. Okay, coenzyme, the non-protein organic part. Okay. And holoenzyme is the complete enzyme containing apoenzyme and the non-protein part. Clear? This is again a repeat of the previous question, so I will not discuss this further. Question 20. A non-proteinaceous enzyme is, and this appeared in 2016. A. Lysozyme, B. Ribozyme, C. Ligase, and D. Deoxyribonuclease. No, the non-proteinaceous enzyme is B. Ribozyme. Okay, so the answer is B. So now ribozymes are a group of RNA, okay, that function as biocatalysts and are non-protein enzyme. Now usually we know that enzymes are proteins, right? But this is a class of enzyme that are non-protein enzymes. Okay, so ribonuclease P is an example which cleaves precursor tRNA to tRNA. And when this ribonuclease P was discovered, they realized that the RNA part of it actually performed the enzymatic activity rather than the protein part. Okay, so there was some protein part, there was some RNA part, but the RNA part actually performed the enzymatic activity. Okay, so performed this function due to the presence of RNA component and not the protein component. So ribonuclease P is a ribozyme. Ribozymes are a group of RNA enzymes, and they function as biocatalyst. Okay, so they are non-proteinaceous in structure. Okay, so question number 21. Which of the following describes the given graph correctly? Okay, this appeared in NEET 2016. A. Endothermic reaction with energy A in presence of enzyme, and B in absence of enzyme. Okay. B. Exothermic reaction with energy A in presence of enzyme, B in absence of enzyme. C. Endothermic reaction with energy A in absence of enzyme and B in presence of enzyme. D. Exothermic reaction with energy A in absence of enzyme and B in presence of enzyme. Okay, so clearly if you look at the graph here, it's the substrate energy is more than the product. Okay, so the product did not really use much energy to be formed, so the rest was given out. So what is it? An exothermic reaction, right? Because it's giving out the energy, it's giving out the heat. Okay, it's a exothermic reaction because the product formed is has less energy than the substrate. Okay, so we know that it's an exothermic reaction, but A with energy A in presence of enzyme and B in absence of enzyme. Yes, that's correct actually. So the correct answer is B. Answer here is B. So A is in the presence of enzyme and this is absence of enzyme. So if you've read NCERT, you clearly know what I'm talking about. You know what this graph is. Okay, basically they are depicting the formation of the conversion of substrate to product and the energy, the activation energy required for it to convert from a substrate to a product, whether it's in the presence of enzyme and in the absence of enzyme. Okay, so now yes, of course substrate would be converted to product. Yes, we know that, but we know that enzymes increase the velocity of the reaction. The rate of the reaction is increased, right? It becomes faster. So this graph actually depicts how much energy is utilized in converting substrate to product in the presence of enzyme or in the absence of enzymes. Let's go to the discussion. You'll be able to understand this clearly. So the graph is given that of an exothermic reaction. Okay, since the potential energy of the product is lower than that of the substrate, right? It depicts the activation energy in the presence and absence of an enzyme. Now, what the hell is activation energy, right? What are we talking about activation energy here? So, activation energy is the energy required by the reactants to undergo a reaction. Now, whenever substrate is converted to a product, if you want to go from point A to point B, you have to move. You have to move physically from A to B, right? So there is an energy that is required for you to cross that barrier. Okay, now substrates need to cross an energy barrier to be able to form a product. That means it needs some energy to move or to convert itself from substrate to a product. Okay, now that is activation energy, the energy required by the reactants to undergo the reaction. Okay, so the reactants when heated undergo a reaction. Okay, but in the presence of enzyme, the reaction is possible at lower temperatures. Now this is a very important point. This is not really mentioned anywhere, but this is a very good point to remember. Okay, so now when substrate has to be converted to a product, okay, they are heated up. Okay, they are all heated up. Okay, and a reaction takes place. Okay, it has crossed the energy barrier in a very high amount of energy, a very high amount of heat is produced, and the substrate is converted to a product. Okay, substrate, if you see, is converted to a product. Okay, there's high amount of heat, that means high amount of activation energy is required for it to cross that barrier. Okay, now if there's an enzyme, okay, if there's an enzyme, this reaction will take place quickly. Correct, it increases the rate, does not change anything else, it just enzymes just increase the rate of the reaction, it just makes it faster. So if you quickly go from if an enzyme is added, it will quickly converted to from a substrate to a product, right? If an enzyme is added, let's say if an enzyme is added here, quickly the reaction will happen. I mean, it's not much heat, will have to be generated to cross that energy barrier, because the enzyme is helping it to do your work faster. Okay, so that means even when there are lower temperature, that means not much heat is being produced, the enzyme quickly converts the substrate to a product, because now it has what, increased the rate of the reaction. You need to quickly go from one place to another. You look for a shortcut, right? When you want to quickly go from one place, from point A to point B, you look at Google Maps or anything, they give you a shortcut. That's basically the job of an enzyme. Okay, they're quickly converting the substrate to a product, so you don't waste so much energy converting a substrate to a product, increasing the temperature. So even at lower temperatures, enzymes play a very important role in converting the substrate to a product. Okay, so if you look at the graph, let's go back to the question. If you look at the graph, look at it this way. Okay, you have a substrate here, you have a product that is formed here. Now for the substrate to be converted to a product, let's say there's no enzyme. Okay, large amount of heat is to be produced. It's like climbing a mountain, so much energy, you're using up so much energy to climb this mountain and then go down and finally form the product. Just imagine the amount of energy, how much temperature would have increased in the reaction. Okay, because of that reason, it's like climbing a mountain and moving from point A to point B, from substrate to product. Now when an enzyme is added, sorry, when an enzyme is added, now you have to move from substrate to product. What happens? It's like creating a tunnel, okay, inside a mountain. So you can move from point A to point B quickly, you can move from substrate to product quickly. That's basically what an enzyme is doing, right? It's creating a shortcut for you, right? Google Maps creates a shortcut for you so that you can quickly reach your destination faster, right? So point A to point B, or substrate to product, it's like climbing a mountain without an enzyme. With an enzyme, to move from substrate to product is like moving inside a tunnel, okay, in the mountain. So now you quickly reach, you don't have to go over the mountain to reach your point B, but you can just move across inside the mountain because there's a tunnel created by the enzyme. So this is what pretty much it is. Okay, if I have to explain this in layman's terms. Okay, yep. So even at lower temperatures, and the reaction is possible because of the presence of an enzyme. Remember I told you this tunnel wala example. Now quickly, you're saving time, right? There you're moving from point A to point B quickly in a tunnel. Similarly, enzymes can perform the reaction quickly, and then you don't need so much heat. No, so much heat is not produced also, and so it is possible at lower temperatures, which is a very good point to remember. Okay, so the role of the catalyst is to lower the energy barrier. We're talking about the enzyme of reactants, hence allowing the reaction to go faster. Of course, we know that, right? So in a biological system, okay, now we have lots of enzymes in our body, and they're continuously, performing all these reactions at a high rate, right? The enzymes increase the rate of the reaction in temperatures below 40 degrees. Now I told you why that rate of the reaction is performed quickly, and so at lower temperatures. Otherwise your body has to maintain a temperature of 37 degrees, right, at all times, right? That's your set temperature in a body. So in a biological system, let's say in our body we are set at 37 degrees, right? The enzymes perform these reactions so quickly that substrate is converted to product very quickly. You don't have so much energy. There's no need for a high activation energy or a high energy barrier to be crossed over from substrate to product. You don't need that high energy barrier, okay, when an enzyme is present. Okay, so this is what activation energy is. I hope you've understood this. You can note this down. These are very, very important points to remember. NCRT also has stated this, has spoken about activation energy. So I thought I'll, brief it down to you, so that's easier for you to now study NCRT. And please remember the temperature part also. Okay, that's a very good important point. Possible MCQ question if they want to make it a little difficult for you. So, yep, answer this question. Let's move on. Oh, okay, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture. I hope you've learned something new today. And please don't forget to like, share, and subscribe to MJ NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. I'll see you in the next video lecture, right? So, bye.